Licciardi?
Sempre al centro della finestra, il microfono vicino. Prima fare una breve presentazione.
Good evening to the present. I am Dr. Licciardi, and I work in the laboratory of biocompatible polymer of the University of Palermo, which is located in the Department of Pharmaceutical Chemistry and Technologies. In my brief presentation, I am going to present the potentiality of nanotechnologies applied to medicine. This kind of application is today usually known with the term of nanomedicine. What does it mean, nanomedicine? Nanomedicine does mean that we will try to treat patients by using nanopharmaceuticals. Nanopharmaceuticals represents an emerging field of drug delivery system, which the most important characteristic is the nanoscale dimension, which is usually compressed between 1 and 500 nanometers. In some cases, nanopharmaceutical can have greater dimension, but anyway, always below 1 micron. This kind of nanopharmaceutical system are therapeutic system composed of at least two components, one of which is the drug, the active agents. The other one are the components of the nano carrier. Nanopharmaceuticals today are very promising drug delivery system because of the finality to optimize drug delivery, bioavailability, and in general increase the efficacy of already known drug molecules respect the traditional dosage form. This kind of implementation can be translated also in the increase of chemical stability of the drug, the promotion of selective drug accumulation and targeting to specific organs or cells, the decrease of administration dose, and with a consequent decreasing in the side and toxic effects, and finally, the possibility to the administration of a different kind of drugs through all administration routes. The importance of nanopharmaceutical in medicine is also demonstrated by the fact that many nanopharmaceuticals are actually in clinical use. For example, starting from liposomes, antibody and their conjugate, vector for gene therapy, polymeric micelles, polymeric drug and conjugates, polymer protein conjugates, and final nanoparticles. In the University of Palermo, our research is focused on the preparation and characterization of new biocompatible polymers, which can be useful to prepare nanopharmaceutical device for biomedical application. In particular, our attention is focused to some synthetic polyamino acids, such as polyhydroxyethyl aspartamide, that we call PEA, and polyaspartyl hydrazide, that we call PI. Both these polymers have a very interesting property for the biomedical application. In fact, they are highly water-soluble, non-toxic, non-antigenic, non-teratogenic, and finally easy to produce in a large quantity and low cost. These two kinds of polymers have been used to produce different nanopharmaceuticals, for example, macromolecular prodrugs, polymeric micelles, polymer protein complex, polycations for gene therapy, and nanoparticles. This is the structure of these two polyamino acids. Macromolecular prodrugs are a particular kind of nanopharmaceuticals in which an active agent, such as a drug, is chemically linked to a hydrophilic polymer carrier, directly or in some cases using a specific spacer. In this case, the hydrolysis of this linkage control the drug release rate. Macromolecular prodrugs so have the advantage in many cases to increase the water hydrosolubility and stability of the linker drug. And the most important advantage is the possibility to target the linker drug to a specific cell. In fact, a particularity of this kind of drug delivery system is the possibility to link to the polymer carrier a specific target group, which make possible the targeting to a specific cell. In our laboratory, different kinds of drugs have been linked to biocompatible polymers, starting for anti-inflammatory drugs, antiviral drugs, and anti-cancer drugs. In the case of anti-cancer drugs, very interesting results have been obtained. For example, the conjugation of paclitaxel, which is a famous anti-cancer drug, to PEA led to a highly increase in the water solubility and chemical stability of this drug. But the most important result was that we was able to promote a passive targeting to liver, thus increasing the efficacy in case of hepatic tumors. In another case, we was able to modify the biodistribution of the some polymeric prodrug. For example, the conjugation of an hydrophilic chain, such as polyethylene glycol, to the same macromolecular prodrug, gave us the possibility to modify biodistribution of this drug, and so decrease, for example, the accumulation into liver. With a consequence, we have the possibility to target the drug to another tissue, for example, by linking it to the polymer a specific targeting group, for example, in this case, the hormone oxytocin. Thus, we promote the selective drug targeting mediated by oxytocin receptor inside the ovarian and breast tumor. Polymeric micelles are another kind of macromolecular, nanopharmaceutical. In this case, this kind of macromolecular system is obtained by the self-assembling of amphiphilic copolymer above a critical aggregation concentration. In this case, the drug is usually physically entrapped in the micelles, but in some cases it can be also chemically linked to the amphiphilic copolymer. Polymeric mice have a great ability to promote cell membrane crossing, thus increasing the efficacy of anti-tumor drugs. In our laboratory, we was able to produce a different kind of amphiphilic copolymer, starting from PEA and Pi. All this copolymer was able to self-aggregate in water and giving polymeric micelles. But important results were obtained by evaluation of the biodistribution of this macromolecular system. In fact, we demonstrated that some of these polymeric micelles accumulate into tumor after parenteral administration, thus increasing the possibility to veiculate a tumor drug inside tumors. In another case, this polymeric micelles was able to increase highly the solubility of very hydrophobic anti-tumor drugs, for example, tamoxifen, and then increase the cell uptake of this drug. Polymer protein complex are another kind of complex in which a physical interaction takes place this time between a polymer and a protein. In this case, the polymer must be properly designed and synthesized in order to be able to complex the protein. And this compensation have to lead an increase of the protein stability. In some cases, the polymer protein complex is able also to promote absorption of protein through epithelial cell. For example, the most important challenge is the possibility to the administration of protein by oral route. In our laboratory, we developed a particular kind of polymer which was able to complex insulin and promote its oral absorption. We demonstrate, in fact, that the hematic glucose level of insulin decrease after the oral administration in vivo of this polymer complex. This invention is now a patent of the University of Palermo, and 2 years ago received a gold medal for invention at Salon International des Inventions at Geneva. Interpolyelectrolytic complex is another kind of physical interaction-based nanopharmaceuticals. In this case, the physical interaction takes place between a positively charged polymer, which is usually called a polycation, and genetic material, such as DNA or plasmid. This electrostatic interaction led to the condensation of genetic material, and with a consequent increase of protection against nucleus degradation, But the challenge is that to improve the transfection efficiency of DNA in vivo. We produce a different kind of polycations starting from P and pi, and we demonstrated that all these polycations are non-toxic and able to complex DNA, but only some of these was able to increase the transfection efficiency of plasmid and thus can be proposed for gene therapy in vivo. Finally, nanoparticles represents the most common example of nanopharmaceuticals. And different kind of nanoparticles exist today, but a particular kind of nanoparticles are polymeric nanoparticles, which are drug-loaded nanomatrices obtained by the chemical or the physical cross-linking of properly modified polymer, which can be synthetic or natural polymers. Nanoparticles have the ability to load high amount of drug and control the release of the drug and biodistribution. We produce a different kind of nanoparticle, and I report just an example of nanoparticle produced starting from PEA copolymers, which was able to escape reticuloendothelial system, thus reducing the non-productive effects and a immunity response. These nanoparticles are in fact loaded with a polyethylene glycol shell, which reduce drastically the macrophage uptake, thus prolonging their circulation time and giving the possibility to veiculate the drug in a specific site of the body. Finally, I can conclude saying that nanotechnologies represent today a very promising approach to design and obtain drug and gene delivery system with aim to solve all the problems connected with the traditional dosage forms. In the laboratory of biocompatible polymers of University of Palermo, different nanotechnologies have been developed and produced for drug and gene delivery, and these nanosystems are today proposed in nanomedicine, diagnostic and cosmetic field. We hope that the research in this field could go on in all the countries of Mediterranean areas and not only, obtaining more and more promising results. Thank you for your attention.
Any comments? Thank you very
Thank you,
much, Dr. Maltese.